Circular RNAs, mostly known as circRNAs, are single-stranded RNA molecules that form a covalently closed continuous structure, mainly deriving from the backsplicing of a pre-mRNA and exhibiting a plethora of interesting features. Most notably, they are widely expressed and particularly stable, since they are resistant to degradation by exonucleases. Regarding their intricate biological functions, circRNAs have been shown to act as microRNA sponges, protein sponges, or protein scaffolds, while there is also evidence regarding the ability of a few circRNAs to encode for peptides. The involvement of circRNAs in cancer treatment response is an emerging field of research with great potential. Several studies have already described regulatory networks in which circRNAs are involved, affecting the outcome of patients treated with chemotherapy and/or radiotherapy. CircRNAs dictate resistance or sensitivity to treatment, primarily through their microRNA sponging function. During this process, a circRNA binds a microRNA and prevents the binding of the latter to its own targets, resulting in the restoration of protein expression levels. Usually, these proteins are members of key signaling pathways and participate in cellular processes known to regulate tumor development, cancer cell stemness, migration, invasion, metastasis, and consequently therapy resistance. For instance, circ-ABCB10 sequesters miR-556-3P, leading to increased levels of adenylate kinase 4, which in turn activates hypoxia-inducible factor 1 and triggers metastasis and epithelial mesenchymal transition. The result of this cascade is the acquisition or promotion of resistance of lung cancer cells to cisplatin. Another example is circ-PVT1, which was shown to confer resistance to doxorubicin and cisplatin. Its knockdown results in a dramatic reduction of the expression levels of the multidrug resistance protein ABCB1, facilitating drug distribution. Furthermore, intriguing aspects of circRNA-induced chemoresistance have emerged. A characteristic case is the induction of therapy resistance through exosome-delivered circRNAs. Exosomes mediate cell-cell communication and intracellular signal transduction, and therefore transfer part of the content of chemoresistant cancer cells to chemosensitive ones. For instance, circ-NFIX and circ-RS122 constitute two pivotal mediators of chemoresistance. CircRNA transfer may result in alteration of cellular properties of the recipient cancer cells, including apoptosis inhibition, increased proliferation rate, and enhanced invasion and migration. Advances in the establishment of experimental protocols are expected to result in the elucidation of the pathways that are regulated by circRNAs and their exact targets, thus contributing to the development of novel therapeutic strategies for patients with solid tumors in order to overcome therapy resistance.